Okay, I'm gonna go ahead and get started here. Thank you all for joining. So welcome to the ISTC Sustainable Seminar Series. I'm Perry Acree, visiting scientific specialist in environmental contamination at the Illinois Sustainable Technology Center. As a land-grant institution, the University of Illinois at Urbana-Champaign has a responsibility to acknowledge the historical context in which it exists. In order to remind ourselves and our community, we will begin this event with the following statement. We are currently on the lands of the Peoria, Kaskaskia, Piankashaw, Wea, Miami, Mascoutin, Odawa, Sauk, Meskwaki, Kickapoo, Potawatomi, Ojibwe, and Chickasaw nations. It is necessary for us to acknowledge these native nations and for us to work with them as we move forward as an institution. The webinar and all these Illinois Sustainable Technology Center's webinars are certified green events through the University of Illinois Institute for Sustainability, Energy, and Environment. To find out more about certified green events through U of I, please visit sustainability.illinois.edu. Just to let you guys know, today's webinar is being recorded. The slides and recording will be available for viewing online within about a week. Everyone will remain muted and questions will be available through the Zoom question and answer feature. You will be able to upvote questions you like within Zoom Q&A using the thumbs up button under the question. I'll be reading the questions to the speaker at the end of the webinar. At that time, the most popular questions will be asked first, followed by questions in the order they were received. If you have any technical difficulties, that's the only reason you should use the chat feature. So today's speaker is Frank Yang. He is an associate professor in the Department of Civil and Environmental Engineering at the University of Nevada, Reno, and is currently on sabbatical at the Department of Geosciences at the University of Tübingen in Germany. His research focuses on the environmental chemistry of organic carbon and emerging contaminants in natural and engineering systems, with implications on the carbon cycle, climate change, and water reuse. He is funded by the NSF, Department of Energy, Department of Agriculture, and Industrial Partners, and his research has led to over 80 peer-reviewed journal publications and 1 book. Frank earned his PhD from Peking University and has been recognized by the Alexander von Humboldt Research Fellowship, the U.S. National Academy of Engineering, the Nevada System of Higher Regents Rising Researcher Award, RSC Emerging Investigators twice, and the EST Best Reviewer Award. Thank you to Frank, and the floor is yours.
Thank you, Perry. So can you see my screen now? So is that in the full screen mode? Sure. Okay, hello everyone. So thank you for joining us. I'm Frank Yang. As Perry just introduced, I am an associate professor in the Department of Civil and Environmental Engineering at University of Nevada, Reno, currently on sabbatical at University of Tübingen, Germany, working with Dr. Andreas Kepler. And it's my great pleasure and honor to be here to share with you some of our recent work about the fate of ionizable organic compounds in soil plant system. And before I dive into the specific topic, I would like to give you an overview about my own, the major research theme and our ongoing efforts. After wandering through various topics, I think the question make me most excited is the organic carbon, their chemistry. This is the thing I have always thought about and always think about when I'm awake in the midnight. So currently we are mainly looking into their redox and the complexation chemistry because it's important for the carbon stabilization, carbon stability, and the carbon cycle on the climate change, as well as the water treatment and the water reuse. The key questions we are pursuing to address in the next 5-10 years is regarding to the chemical structure of these unknown reactive components in this complex media and their reactions, including the reaction thermodynamics and the kinetics. And we would like to understand the carbon stability under the different climate change scenario, include the impact of the wildfire, and also apply this knowledge to develop the amendment technology to stabilize carbon in the permafrost soil, as well as to treat the contaminated water for reuse in agriculture and other scenarios. So following this, I would like to give you a bird's view about like our current ongoing efforts. Like my students, like the Sri and Anu, have been working on the application of the biochar for dehydrogenation and degrading of the organic pollutants. This is an NSF project where we work with colleagues from University of Minnesota and Georgia Tech. And in Shri's first work, we have demonstrated that the biochar in the aqueous phase, what we call the aqueous biochar, can dehydrogenate organohydrogen compounds. Based on the electrons, like the calculation, it's even similar as reactive as nano-sized zero-valent iron particles. So the current bottleneck for the large-scale application of such technology is the solubility of biochar, so which we have been working on. I will mention some relevant effort towards the end of this talk. And currently, like Anu and Shri have been moving forward to study the chemical nature of those reactive components, which is mainly quinone in this complex media. And with this knowledge about the biochar, we also apply that to study the impact of the wildfire on the watershed function, because the materials you can observe after the wildfire, including those like the black like ashes like on the surface of the soils, which I can observe when I drive through our Sierra Nevada mountain crossing the Nevada-California border. They are pretty similar to the biochar we have been using in the lab, like for the engineering application and the dehydrogenation reactions. We have been looking at how the carbon and the nitrogen are mobilized after wildfire, with implication and the protection of our precious water in the Lake Tahoe, which I have been observed as the most beautiful and precious water resources. For this project, my student, like the PhD student, like Travis from our like the local tribe community has, and other like undergraduate student Justin have been like analyzing these materials collected from the wildfire around the Lake Tahoe region. So this is our like fundamental project about the quinones. And we have been also encountered another very specific quinone, which is titled as 6-PBD quinone. If you Google this compound, you will find lots of the news, like after December 2020, like one of my previous colleague, Dr. Ecology at University of Washington, has discovered this specific compound as a killer for the coho salmon in the Northwest region of United States. Like it caught like lots of the industry under the like attention, including my colleague from the pavement engineering, where they have used the tire rubber as amendment for the pavement industry, one of the major like reuse for the tire rubber. But to sustain like their like environmental friendly reuse for the pavement materials, we need to understand how such compounds can be released from the rubberized pavement materials, how they behavior at the specific interface of the rubberized pavement and water. And I also perceive like this very specific interface as a very unknown area for the stormwater management. There can be lots of the interesting compound and the behavior occurring at this interface. So these are our current efforts about the redox reactive organic matter. And to the other line, we have been also looking at the complexation active compounds, such as the ligands. For that, we have been like looking into the organic ligands generated from the lignin degradation with important application for the carbon stability in the soil environment. For this project, we have been working with Dr. Rene Boato from Oregon State University to develop a protocol to identify the unknown ligands generated from the lignin degradation, where we can use iron isotope pattern to identify those suspects and identify their chemical nature by the high resolution mass spectrometry. And my new student, Alisa, also joined these efforts. So like such like organic ligands and their interactions with the minerals play important role for not only the carbon stability, but also the fate and transport organic pollutants. So for that, we just got another project started this summer at Alaska to collaborate with University Arizona, University Alaska, and the local tribe community on Alaska Island to look at the mobilization of organic pollutants derived from the formerly used defense sites and their transfer to the local subsistence food and their threat to the human health. Yeah, for that, hopefully we can use our knowledge about the carbon like mobilization to understand how these organic pollutants can be transferred to the food chain and finally end up into the human exposure and how we can mitigate that together with like the stabilized carbon under the climate change for this like the very sensitive region. Like regarding the organic like pollutants like feed transport, we have been just finishing up like a NIFA project regarding to the feed and the plant uptake of the emerging organic compounds, for which will be my major topic for today. Like my student Abra is also the major driving force behind this project. This is a NIFA project led by my colleague and also current chair, like Dr. Krishna Pagila. For the overall project, it including the modeling components and the greenhouse culture and also the field study, like leveraging our own like university agriculture farm station with focus to understand the human health impacts from the emerging contaminants in the reclaimed water for the irrigation use in the agriculture section. And for this topic, like maybe like many of you have already been aware that the agriculture use is one of the major sections for the water withdrawn like in the broad region. So this is estimation like conducted by the USGS, where you can see that like 40% of the total water drawn is for the thermoelectric power. Under that, the major section is agriculture, including the irrigation, then the livestock use, and also other like aquaculture use. So that in total contribute around like 40% of the total water we withdrawn. So that means like to develop a sustainable management of the precious water resources, the agriculture is an unavoidable like the topic. That's the reason there has been lots of efforts on the water, like the food agriculture nexus. There is a great potential to reuse treated like wastewater effluent for the agriculture irrigation, because our current technology can make those water to the very high quality, even like so we can even start developing the direct and indirect potable reuse. But like as we develop the technology and knowledge about the chemicals in the water and the wastewater, especially those in the wastewater effluent, we are aware there are a huge amount of the different kind of organic compounds in these like the materials. This is a survey study conducted by our colleague and also friend like Dr. Benny Chaves from Israel. So this is a large like the screening study for the organic compounds occurring in the wastewater effluent used for the agricultural irrigation. So the axis here is a different like chemicals which we can capture, like where they mark the detection frequency with the concentration. So that means the residue concentration can range from like the nanogram per liter to microgram per liter, with some outliers up to like milligram per liter. And based on the different groups, we can also see their detection frequency with the average concentration and other statistics. So that means like to ensure the safe reuse for the agriculture irrigation, we really need to understand how this a large number of the compounds are processed in the soil and taken up by the agriculture product and ended up in our diet, and also how it leads to some potential human health like the risk, and how we can mitigate that. So that's the reason we have been developing a screening model to prioritize the emerging organic compounds occurring in the like wastewater effluent and how they can transfer through the agriculture crop and to the grazed animal, like which is like beef product here, and to the human exposure. So our modeling, the major results are based on our own, like the University of Nevada, like agriculture farm, which has used the reclaimed water for irrigation over the 20 to 30 years in the past. And it gave us great leverage to work on. But by the end, we also expanded this screening model to a tool where we can prioritize compounds based on their physical chemical properties to prioritize certain kind of compounds with a high risk or high chance to mobilize and transfer to cause some human exposure. And based on the detection frequency and the concentration, as well as their human health concern, we have used 5 target compounds as model chemicals in our screening model development, including the triclosan, naproxen, and other compounds we listed here. And to address their like the soil fate and also plant uptake, we have developed the MATLAB coding to cover their like the absorption, desorption in the soil environment, then their leaching and the degradation, and then the bioconcentration factor from the soil to the plant. And consequently, the bioconcentration from the agriculture, the plant, which is alfalfa here, to the beef. Then, based on the statistics for the human, like the behavior, meaning the dietary data, we can estimate the human exposure to these organic compounds through the food exposure. And finally, we can do the risk assessment for these compounds. And this is the screening model. We want to make it relatively simple. So that means the stakeholder and the manager can use it in the relatively easy way. So this is our major like the processes. Like we have the data input from the field survey and also include the data we measured, including some data we collected from this overall NIFA project, including what my colleague Dr. David Hannigan has measured and investigated for the concentration of these emerging contaminants in the wastewater effluent and including their processes and the concentrations after different like the treatment steps, so we can understand like which process is critical for removal of these compounds from the wastewater like the perspective. And from the results, this is time-dependent concentration of these compounds, and in the soil, which is shown in the left, and also in the plant, like shown in the like right axis, and axis is the time, and where we have the 5 panel for the different compounds. And the solid line here is for the soil concentration. The dashed line are for the plant concentration. And the blue line here are for the average, and other lines are for the percentage, like the numbers. So overall, we can see that it took around 100 to over 500 days for this compound to approach or achieve the steady state concentration in soils, depending on their physical chemical properties. Like for triclosan and naproxen, it can approach the steady state like the relatively short within the relatively shorter period. When the carbamazepine and the fluoxetine, the time reach the steady state is a little bit longer. And also we can see the average concentration of these compounds like achieved after they get into the steady state. So that's a concentration we have been using to calculate the consequent concentration in the plant, and then the beef product as well as the human exposure, where we resulted the human exposure to these different organic compounds based on the different age group, because the assumption of the consumption of the beef is different. And we have that data, like then we can calculate the human exposure through this wastewater irrigated agriculture, like the chain. And the big picture message here is that if you really look at the value we calculated for the human exposure, they are orders of magnitude lower than the acceptable daily intake of these emerging contaminants, like the recommended in the literature. So that means, like for example, for triclosan, the recommended number is close to 10 to minus 1 milligram per kilogram per day, when our calculated exposure through this way is 10 to minus 10, like even below that, milligram per kilogram per day. So which means like for each of these single compound, their risk is almost negligible based on our estimation here. But several notes we need to like the take is like this is as we show in the beginning, like in our wastewater effluents, there can be like thousands of different emerging or even some unknown compounds. So that means when we look at the single compound, their risk is very low. It doesn't mean the overall risk is necessarily also very low. And there are all still like lots of the debate like going on in the study or in the community about like what level of the risk really caused by this like wastewater effluent. And secondly, the input concentration of these compounds are based on the concentration we collected from our local treatment facility, which is an advanced facility, like where we include the primary, secondary, and tertiary treatment, like including the sludge treatment, like the nitrification, denitrification, and also the chlorination. So that means like we also have our concentration compared with the nationwide like the advanced treatment effluent. So but for some small facility or less like the centralized treatment facility, the concentration of such emerging contaminants like can be higher. But overall, like for the scenario we are looking at, the risk raised by this like the compounds are quite low. And then if we want to expand our calculation to even a large range of the compounds, because we're only looking at like 5 compounds here. So we have expand our calculation to a screening tool where we can map the different kinds of compounds in this like analysis, where we have calculate index, which we call the reuse transfer efficiency. So basically we get the concentration of this in the compounds in the agricultural product, which is beef here, over the concentration of the compounds in the effluent. So that means the higher of this number, which means these compounds are more possible to be transferred into the agricultural product and consequently called the human exposure, and which is shown in the color here. So that means the more yellow, it means a higher number, the more blue, it's like a lower number. We like the place this calculation against their physical chemical properties of these compounds, including the KOW and the other ratio of the degradation rate constant over the bioconcentration factor. So that means the higher KOW, then the lower degradation rate, or the higher bioconcentration factor, like the lower degradation rate, a constant, the transfer efficiency would be lower. The higher KOW, the lower degradation rate, this like transfer efficiency can be higher. Yeah, sorry for some confusion here. So that means if we are looking at the compounds we have studied, like the triclosan and a couple other compounds are on the higher end of this, like the spectrum. One other compounds, including the ciprofloxacin, is in the lower end of the spectrum. So that means we can prioritize the compounds for the further scrutinize. So that means like the compounds with certain physical chemical properties can have the higher tendency to be accumulated in the agricultural product and potentially cause some human exposure. So this is what we have collected like from the screening level modeling study. So where we demonstrate that the overall, especially the risk coming from the single compound, especially for the 5 target compounds we have specific modeling, they are relatively low or almost negligible. But we can also use this tool as a screening like the process to prioritize the compounds for the next step, like investigation. If we really look at the compounds we have been studying, such as fluoxetine here, we will notice that these pharmaceutical compounds, they normally will contain some functional groups such as amine group here. So like these amine groups, they can protonate, deprotonate depending on the pH and the pKa of the related compound. So the charged species can have the complex interactions with the soil minerals and organic matter through the charge interactions, like ligand exchange interactions and other like the Van der Waals interactions, and also the aromatic like ring-based interactions, such as these aromatic rings also in these fluoxetine structures. So such functional groups are pretty common, especially for these like emerging or pharmaceutical and personal care products, because these functional groups are important for the solubility of such chemicals, which can make them efficient in use for the drug or other personal care. But these complex interactions between the ionized, like the species of the compounds and the soil components, are almost like ignored in the study, especially the modeling study for the plant uptake and soil processes of these compounds with contents for the agriculture water reuse. So that's what we have been working on as a following study from the first screening, like the modeling, like where we have developed this small, like the process-based model where we section the plant into the root, stem, leaf, and fruit, and where we consider the leaching, evaporation, degradation in the soil, and then the uptake by the plant, and also the internal, like the transfer between the different tissues in the plant, as well as other like removal, like the reactions. For that, we have used like the two typical agriculture plant, like lentus and soybean, for our study. And we will mainly show one of the results, because like all our results are quite like the complex. And with three like emerging contaminants, including ibuprofen with a carboxyl group here, and fluoxetine with amine group, and the triclosan with a phenol group, which cover the most common like the ionizable functional group, which can occur in these like organic compounds. And to like really address their like the processes and interactions between the neutral and the ionized species with the soil minerals and organic compounds, we have developed the modeling based on the different scenarios. We started from the very basic scenario, which is similar to our the modeling, the first screen modeling, where we do not consider the speciation. We only use one like parameter such as the KOW to estimate their absorption and the partitioning. Then we move on to the scenario 2, where we consider speciation, but in a very simplified way, where we only assume the neutral components of these compounds can be solved by the soil. Then we ignore the absorption of the charged species, which is idea like the behind the DOW, which has been used in the literatures. And then we move on to the scenario 3, where we consider the both neutral species and the ionic species for their absorption by the soil, but we only consider the contribution of organic matter for the absorption. We do not consider the interactions with minerals. Then finally, our scenario 4 is the most complex one, where we accounted for the interactions between the neutral and ionized species and their interactions with both soil organic matter and minerals. So as you can tell, our scenario 4 is the most comprehensive one, where we are using as a benchmark to evaluate the interactions with such ionizable organic compounds with the soil in this like water reuse processes. So to compare the scenario 4 with other scenarios, we can elaborate how the different like the components of these processes contribute to the fate and plant uptake of these compounds. For example, if we compare the scenario 4 and scenario 3, the difference can elaborate the contribution of the minerals to regulating the plant uptake of these compounds, for which we'll elaborate the results in a minute. And to calculate that, we need to use relatively complicated like the equations. I promise this is the last slide where you will see a couple equations for calculation. So for concentrations, we have used this like the differential equations to address the root soil root transfer, then the leaching, then also the volatilization and the degradation. And for the concentration in plant tissues, we address or we counted the internal transfer and also the growth dilution. And more detailedly, to address different components and their interactions with soil organic matter and the minerals, we also use these partitioned like the equations to account for the interactions of the neutral and the charged species with the soil organic matter and minerals. And we use, for example, this equation to estimate the sorption of the organic compounds by the organic matter in soils, including both neutral and the charged species. So for the details, you can track the recent publication by Albert. So let me know if you have the detailed questions regarding that. And based on this, like we can do the calculation. So we can see that it will take like the 200 to 400 days for the concentration of the compounds like to approach the standard state. So for here, we show the ibuprofen in the scenario 4 for one plant, which is the lentus here as an example. So the detailed results are quite like the complex. You can find all the data in our publication. So then the other like the important like the notice is like when we look at the different pH, which is a pH 5, 7, and 9, we notice here, if you look at the soil concentration, there are not like much difference. So that means like consider the speciation or not consider them is not that really important for calculating the concentration of such compounds in soil. But it's not the case for the plant. So when we can see like like quite big like difference when we change the soil pH from 5 to 9, which is a normal range which you can find in agricultural soils. And you can also notice a dip like for the plant concentration. This is because like we have calculated like the plant concentration considering the harvest cycle. So that means after each cycle, the concentration in the plant will start from 0 again, but the soil concentration will continue to accumulate when we assume we start from a clean soil with the input of these compounds from the wastewater, like the effluent irrigation. So we can also see like for the different like tissues, their response are also different. Like based on because we have like many parameters included in this calculation. So before we further scrutinize their results, we also want to understand our like error bar or the uncertainty for our calculation. So for that, we have like collected the data available for some critical parameters included in our degradation in our calculation. Then we can do the Monte Carlo simulation, which Abro has done very hard work to conduct like thousands of the calculation, where we can get the standard deviation for our calculated results of the concentration. So this is the data like where we can see the like solid line in the middle is the average result when we have this shadow indicated the error bar where we collected for the concentrations of for this one we use fluxing as a model for the soil and also different tissues of the plant. And to tease out the contribution of the different parameters to this uncertainty, we also have done the sensitivity analysis, where we show on the right side, like where the y-axis is the concentration and axis is adjusted factor we have applied to the parameter. So that means when the degradation rate constant is adjusted from like 0.1 to like to 1, which means we like change the concentration by multiply with these factors, we can see the concentration has a significant or substantial response to that. For which we will also elaborate the reason like in the following discussion. And so now we have a solid understanding about our calculation. So from there, we can really see how we understand or what we really like to know about the pH and impact of the speciation of these organic compounds for their calculation. So this is one of the probably the busiest slide like I'm showing here. So it's showing the concentration of the two like compounds as example. One is the fluoxetine, the other one is the triclosan, and we also show the two like plants as example. And we show the concentration from the soil and also the different plant tissues here. And then the different, we also show the different, like the pH from the 5 to 9 as a different group here. And also the different color showing the different scenario from the scenario 2 is a blue bar to the very right bar as the scenario 4. And the scenario 1, because it doesn't have any speciation, so which is a constant across the pH, which is shown as a dashed line here. So from this like BZ calculation, like there are several important like the knowledge we have gained. So as we said, like in the beginning for this calculation, we can use a scenario 4, which is very right bar here as our benchmark, because we think this is a calculation where we comprehensively consider the interactions with all the species with the soil components. If we compare that with other scenario, for example, the scenario 1 and scenario 2, we can see that for the soil concentration, the difference is not that like the substantial, especially for the triclosan. So that means the consideration of the speciation and the pH are not that important for soil, which we'll also explain in a minute. But it has a dramatic or drastic impact for the plant concentration. So that means like when we use the scenario 1 or scenario 2, especially there's no like the consideration for the different speciation and their absorption by the organic matter and the minerals, the difference can be like the several orders of magnitude or up to 10 orders of magnitude compared to the final of scenario 4, where we consider all the interactions. And like the scenario 2 is even the worst. So which means like when you assume the neutral speciation is now solved, which is a case where we use this concept of the DOW. It probably even made like the more like the miscalculation for the plant uptake of such compound, because there has been like the many studies which shown the interactions of the charged species of these compounds with the soil, organic matter, and the minerals, because there can be lots of the interesting interactions based on their like the charged functional groups. And then when we compare the scenario 3, which is this green bar and the very right bar, we can see the contribution of the mineral, because for the scenario 3, we only consider the contribution of soil organic matter. When in the last scenario, we consider both organic matter and the minerals in the soil to the absorption of these compounds. We can see such like the mineral contribution is dependent on the compounds, also dependent on the pH. For certain cases, such as in the soybean evaluation, like for their fruit, like the difference is relatively small. But for the triclosan in the lentils in the leaf, which is like the consumable part, like the mineral contribution is like up to like over 1 order of magnitude. So that means to consider the contribution is mineral of the mineral is also very critical for such evaluation. So that means hopefully like through this calculation and like demonstration here, I have convinced you that like the first, it's very important to consider the pH and as well as the different speciation of these compounds for the plant uptake calculation. And the second, like it's the mineral contribution or the different components of soil, their like the contribution is dependent on the compounds. And the third, it's quite different for the soil and the plant. For the soil, such consideration seems not that important. So why? So to understand that, we need to look into the fluxes. So that means because we are doing this dynamic calculation, so that's if you imagine out our differential equations. So for the soil, there are many like the removal processes, which is shown here, including the leaching and the soil root transfer, evaporation, and degradation. And similar for other components, such as for the root, it's a 2-part, for stem it's 3 parts, and for leaf, it's a 2-part. And for the soils, these 4 parts, like the what we call the efflux, are showing as this pie chart in the like bottom left here. But you can see that for all these pie charts, they are dominated by this part, this green part, which is degradation. So that means like degradation is a major like efflux for the soil concentration of these organic compounds. So that means like when we consider their dynamics, this degradation like the importantly regulate their steady state concentration. But for these degradation processes, there's very limited knowledge or almost like the rare data regarding to the degradation of the different speciation of these organic compounds, which we perceive as a very important knowledge gap to fill. So that means in our modeling, we do not have the different degradation rate constant for the different like the speciation of the compound. So that's the reason I consider that are not important for the soil concentration, which kept constant in the different pH. And the consideration of the speciation do not bring too much like difference for that part. But we from this calculation, we think like to understand the availability of the charged like the speciation of these emerging like organic compounds and their consequent microbial degradation is very important to like to fully ping out the picture for their plant uptake. And then we also want to expand our like the calculation to a large range of the soil conditions. Like the one, our major calculation is still based on the a typical like the agriculture farm soils. And so we have developed a similar like contour map as in our first screen modeling, but here we evaluate the uptake factor for these three different compounds. Which is calculated as a plant concentration versus the soil concentration. Like like we map that against the pH and also the soil organic carbon like the fraction. So that means like if we look at the color here, the more yellow color means a higher like uptake factor. The blue color indicates lower. And we also put the data we can collect from the reported literature where we can find the data about organic carbon content and all the pH available. So for example, this triangle indicates the reported like uptake factor for ibuprofen is 1.8 to 3.2, while our modeling calculation is in the range of the 1 to 1.5. So that means it's comparable as we also showing in the bottom, like the figure here, where our calculation is the blue bar is experimental data, like the orange bar is our calculation. So it's not ideal, but like at least give us comparable like estimation for the plant uptake factor. So that means like so we can expand our like the prediction for the plant uptake of such like the ionizable organic compounds in a larger range of the soil chemistry condition, including their organic carbon content and the pH range. So that means like through this complex calculation for the ionizable organic compounds, I think like we found out the speciation of such compounds are very important for to be considered for calculating their like the plant uptake, although not that important for the soil concentration. Although we need to know that this is based on the assumption that the degradation of the charged component and neutral component are the same, for which we believe is a major knowledge gap for the next step studies. And from this study, hopefully you are also on the same page as myself. We can also very easily see that the interactions of the organic matter and the minerals can play some critical role in regulating the fate and plant uptake of these emerging contaminants, because those compounds can have a very important interface for the emerging contaminants to stay with. And to understand that, which is our current like the major efforts to really try to fish out the complexation reactive organic compound together with the redox reactive compound. So because like when you inject the normal organic matter materials into the mass spectrometry, you normally will get a smear peak like what I show in the middle here, which include like thousands of the single feature of the peaks, which represent like thousands of the single components. So for us, our efforts is hopefully in the next 5 to 10 years, we can build up a library to screen out among these thousands of compounds. Can we list out the top like a dozen or dozens of the most important components among that for the redox reactions and the complexation reactions, which we believe are most important for the carbon stability in terms of the carbon cycle and also for the emerging contaminants like the behavior with the contents for the water reuse. So our strategy is to couple the chromatography separation, then the reactivity screening, as well as the chemical tagging and high resolution mass spectrometry analysis. So we think that we normally will get the organic carbon materials like through the extraction, then we can concentrate them. When we get the materials like sufficient for our analysis, we'll do the consequent like the column separation. Like so hopefully we can separate them to the degree where we have the manageable number of the compounds as our suspects or the candidates for the further analysis. And for those like the small pool, the compounds will do the reactivity screening analysis. Then we can identify the highest or the most important fractions for our next step analysis, for which we will do the chemical tagging. So the idea is to give a chemical label for the suspects we are concerned about to be identifiable by the high resolution mass spectrometry. For the complexation compounds, we are using the metals, which means is iron, as a tag for them. So that means when they complex with iron, we can use iron isotope of the 56 and 54, which is a natural abundant, to screen out the suspects for our next step analysis. Then when we identify those candidates, we can do the second MS to get their fragment and coupling with other the analysis such as NMR, like the spectral analysis, to get the structure. Then hopefully we can use the model compound to validate that. So this will be a really comprehensive way we can build up a library for the compounds which are still unknown in the natural and engineering system, which is normally perceived as a black box. And hopefully in the same time, we can have other lines of evidence to support our analysis, such as for the metal complexation, we can use the ICP-MS and the ICP-MS to really verify the complexes we captured. And this is our ongoing the efforts to give you some the preliminary idea about what we have been doing. Abra has also done lots of work to generate the media for the lignin degradation by incubate the lignin with some bacteria which is demonstrated to be able to degrade lignin. Then we get the media, which including the materials which generated from the lignin degradation, which represent the materials you can encounter in the natural soils after the biomass degradation. So these lignins are claimed as the most important, the macromolecular and also the most stable and complex organic compounds in the natural environment. And based on their structure, it's also believed to be able to generate a range of the different organic ligands, which is still unknown to the community. So after we get these materials, we will concentrate them to give us sufficient material to play with. Then we will add iron to it. Then hopefully it forms some complexes. Then we do the column separation. Then we can analyze iron content to prioritize the components for our next step analysis. Then we can really use an isotope pattern to identify the suspects or the complexes or the ligands we want to further investigate. Because for the iron, we have this 56 and 54, where they have this the difference in the MZ, which can be measured in a very active way by the high resolution mass spectrometry. And they also have these intensity ratios by their natural abundance. From this, the range, we can narrow down the possible candidates for the complexes in our materials to be the further analyzed for their structures, for which we are using the MS1 to identify their molecular formula, and also the second MS to identify their structures. So this is MS1, this is MS2, where we get the screening analysis. And we can also use metabolomic the tools and the protocols to build up the fragment pathways. But when we look at this the complex media, it's very critical for us to ensure our understanding about the MS analysis of these complexes are really true. So that's the reason we also developed the bottom-up strategy, where we have been using the model compounds from representing the lignin degradation media, including the caffeic acid, the cumuric acid, vanillin, and cinnamon acid, which include or are composed of the functional group such as the carboxylic and the diphenyl group, which is also called the catechol groups, or just one phenyl groups, or one phenyl with ether groups, or just one carboxyl groups. And from our high resolution MS analysis, we can determine their complexes which we can capture under this like high resolution mass spectrometry, and which is quite different from what can be predicted from the traditional like the aqueous chemistry or some titration experiment. But we need to know that when we do those experiments, we need to assume the species like formed which are only identifiable in the MS analysis. So there's still like a knowledge gap, like the community need to understand to really bring these two parts together. But this analysis can give us a library and how we can really understand the complexes in the more complex media, such as from the lignin degradation. So that means whether it's a caffeic acid like complexes, which means they can form the complexes such as this way, including the mono ligand like monodentate or monoligand bidentate or biligand like the bidentate like complexes, or it's cumuric like acid like type ligand or complexes. So by the end, we will group our unknown ligands in the different ways, and to address the other challenge for the complex analysis for which we do not have the standard to be either available for other like organic compounds. And like finally, I just want to very quickly, yeah, these are the second MS we captured for these complexes, and also the fragment like the pathways. For the sake of time, I'll also want to just briefly mention that we have been also developing the chemical tagging for the quinone groups, which is also difficult to like the chemically like label, like, but we have used a micro addition between the cysteine and cysteine, like the base, like the peptide to label them. And Anil and Shri has also developed method which we can use to quantify the quinones in the complex media. So if you have interest for this, our major efforts, like for now, like hopefully I can update like with you in the near future. So we think that hopefully I have given you a comprehensive like the discussion about what we have been interested about, with a case story about the fate and transport of the emerging contaminants in the water reuse. And hopefully you are on the same page now with me, that like to understand the organic carbon structure are very important for the carbon stability, for climate change, and also for the water reuse. And my colleague and I have published and edited this book, like just published this year by Wiley, like for the multi-scale like biogeochemical processes in the soil. And hopefully it can serve as a reference textbook for the community. And last but not least, I just want to acknowledge all the like funding resources we have received, including multiple programs from National Science Foundation, NIFA, and other industrial partners currently. And we think that like thank you for your attention, and any questions.
Excellent. Thank you for a good talk, Frank.
Thank you.
If anyone has any questions, you can put them into the Q&A chat function, and I will read them out.
Yeah, for my last slide, these are just some photos from our Tahoe Lake and also our new engineer building at our campus, which finished before the COVID, which make everyone released, and other some like a hot balloon events in our community, and also some ski resort. So hopefully I can welcome some of you in our Reno campus in the short future.
That's beautiful. I have a question, or I can get us
Sure.
started really quickly. Do you have any idea through your research so far of, I guess, is there a certain between like or is there a certain tendency for some of these emerging contaminants to accumulate in either like plants or like in different parts of the food chain, like plants or the animals that consume them?
Yeah, that's a good question. Like in our first modeling study, I didn't show the details, but it can be found in the paper. And we have calculated that transfer efficiency for the agriculture plant, as well as for the grazed animals such as beef, which we calculated there. So for the because like for the beef, there is additional step for the bile concentration, which people have developed a regression like equation to estimate their bile concentration factor. For the plant, it's mainly like uptake from the soil. So there are the different, they are largely like a similar group of the compounds, which is normally the higher like the partitioning coefficient, which will make them return in the soil. And if it's a lower degradation, it will probably make them higher tendency to be transferred to the next step. But there are still some difference because like there are the additional steps there. But yeah, for example, the triclosan is some of the compounds which is among the list of the compounds which is more likely to be transferred to those like the end products from agriculture. But for that screen model, we have used relatively simplified like the calculation to be used as a tool for the screening. The one in our following study, we have been using like more comprehensive like the processes. As we elaborate, we consider like the different parts of the organic compounds and also their interaction with soil, and even sections the plant to the different tissues. And there are still lots of the unknown like the parameters, including the soil degradation, like rate constant, which is still one of the major knowledge gap, as I said in the talk. And the other part is about the processes of this compound inside the plant, how they are internalized, how they interact with proteins, and how the different species within the plant tissues, and how is their availability. So there are still lots of the interesting questions regarding to those like important parameters. Yeah, so hopefully that makes sense to you.
Yeah, did. Excellent. Looks like we got a couple questions in the chat.
Sure.
First one, have you looked at the perfluorinated compounds and if they behave similar to PPCP compounds you studied? You knew that was coming, right?
Yeah, that's a very relevant question. Like that's some of our targets, because when we are doing the biochar like a dehydrogenation, so that's some like our emerging enemy like for our application or the biochar. But so far we didn't have the opportunity to really use our biochar to fight with organofluorine. But we think based on the reduction potential, those biochar can be potentially used for degrade some of those organofluorine. And for this, like the plant uptake, like the processes, like we didn't like including the organofluorine in this part of the work, but that's something like within our like the original plan, but we didn't get chance to look that far. And so far we only look at other compounds, which is more common in the wastewater, and also with a more reliable like the pK values. But for most of those, like the fluorinated organic acid, there is still some debate going on about their pK accurate values. And based on the current value, most of them are just stay in one of the species in the most like the agricultural soil pH range, which make them less interesting to be calculated in the current like framework. But that's definitely like some interesting thing to think about. But also to the other side, yeah, those compounds are not really that high concentration in the wastewater reuse, and also they have the higher tendency to be like absorbed by the soil minerals and organic compounds. So that make them probably I speculate they probably have the lower tendency to be taken up through our modeling calculation, but we didn't do that far. But that's definitely something we have been think about. Thank you for that question.
Okay. Can you comment on physiological processes of plant membranes, or are you only looking at steady state? Also,
Yeah.
heat and humidity, is that affecting the transpiration rate?
Yeah, that's also a very relevant question. Like because as you can tell, like there's when we do this modeling, I guess there's a tricky boundary where we can make the modeling still manageable, but still comprehensively address all the components we can consider. So that means like for the plant tissues, like in our second part, which is more comprehensive than our screen modeling, we have sectioned that into the root, like the stem, leaf, and the root, like the fruit, and we have considered those internal, like the transfer, but we didn't consider those like membrane, like the processor, which is probably also important for the uptake of these compounds. And there are some relevant study in the literature where they have like developed those section models, like considering the membrane processes for the uptake and internalization of the different species of these organic compounds. But that's like very important and interesting, like the parameters to further develop. But for right now, our current modeling, we are using the steady state as our baseline for the human exposure estimation and also the evaluation of the importance of the different species and the soil pH.
Oh wait, sorry, I was muted. Okay, last question. Your results to date seem to present little risk to humans from using wastewater for irrigation. Does this make it difficult to get further funding?
That's a good question. So that means like from our scientific perspective, yeah, maybe the numbers are not that interesting when we see the very, very minor risk. But I guess that's good for like the real world application, right? Like if we want to promote the agricultural reuse, these results are meaningful. But as we stated in the discussion, like we still need to acknowledge that there are a large number of the compounds existing in the wastewater effluent. And without like talking about those unknown compounds, we still do not have a chance to elaborate. Like we are inputting like the tens of thousands of new chemicals into the chains for these like the processes. So that means like I guess this like monitoring and the understanding their behavior in the wastewater treatment processes, as well as how they process in the agricultural soil and uptake by the plant is still very important to secure the like agriculture, like the reuse. Like we have the interest and the development in like put this into the framework for the biosolid application. So because for the biosolid, they have the higher concentration of these contaminants because they can sob and reserve lots of those hydrophobic organic compounds. And when they apply to the agricultural soils, it's also important to look at their fate and plant uptake. Thank you.
Okay, awesome. Yeah, I think that's just about our time. So once again, I'd like to thank Frank Yang for a great talk, and thank you all for participating, and we'll see you next time. Thank you.
Thank you.